Takedown. This is a response to Dr. Jay Bhattacharya and Dr. Matthew Mamoli's essay in City Journal, in which they sort of talked about what they think a pandemic preparedness plan might look like. They didn't. And we're going to talk about that. But as people file in, I'm going to just repeat all of that. I'm Jeremy Faust. This is the Doctor's Lounge. And that's the Substack Live of Inside Medicine. And I'm joined by virologist and general badass, Dr. Angela Rasmussen.
Hi,
How are you?
everybody. I'm great. How are you doing, Jeremy?
I'm
Dr.
fine.
Faust.
Yes, Dr. Rasmussen. We'll drop ranks in a moment, but Dr. Rasmussen, we did, I think, kind of light up a little corner of the internet, of our internet over the weekend with a response, this rebuttal that we wrote. We wrote a rebuttal to the NIH leadership's screed against pandemic preparedness. And just to set it up for everyone who may not have seen it, the NIH director, Dr. Jay Bhattacharya, and his deputy, Dr. Matthew Mamoli, wrote an essay in City Journal, which is the Manhattan Institute's journal, about what they call it "The World Needs a New Pandemic Playbook." And the subheader for that was, "The old one failed to cope with COVID and may have even caused it." So we know where this is headed from that sort of gauntlet, which is why I reached out to you, Dr. Rasmussen. I think a place to start is actually something that sometimes gets skated over. And that is, to the best of our knowledge, where did this pandemic, the COVID-19 pandemic, begin? And I ask you, not randomly, I ask you as a very impactful co-author on some of the most important science on this topic.
Yeah, so this is a great question, and it's one that never seems to come up anymore. This one of the things that was so irritating to me about this article was that just said may have caused it, but it's really a presumption that it did cause it, that it was the result of a lab leak. But the evidence actually shows otherwise. And the evidence shows that the pandemic probably started at the Huanan seafood market in Wuhan, China, in affiliation with the live animal trade. I'm happy to go over the
Yeah.
seminal papers that describe that.
One
So
quick thing. One quick thing. Sorry, your camera's kind of shaking. I'm not sure if it's like on a.
it's because I'm holding it.
Oh, do you have like a book or something? Because it is a little bit.
Yeah,
Uh,
I'll try to keep my hands
I
still
don't want to
because
make everyone, think that we're at sea. But anyway, especially because your words are very important right now. And I'll just say that there's some papers, and again, our top journals that you were involved in. And when I read these papers, I had nothing to do with them. I just read them as a consumer of science. My jaw hit the floor because you literally have like a map of the market, like where you can buy this kind of raccoon dog and this kind of animal, and it's really a floor plan. And you've got genetic sequences from specific cages, correct?
well, not specific cages, but specific stalls and locations in the market.
Okay, and from first of all, how did we get that data?
So this is kind of a long story, but I'm going to try to make it quick. We got this data through a series of different sort of leaks and happy accidents. Initially, we didn't have any of this data. So to kind of go back to the very early days of the pandemic when people started to wonder if it was a lab leak, one of my colleagues and co-authors, Christian Anderson, was looking at the sequence of the virus in late January of 2020, and he said, "Well, this looks pretty weird to me." And he's an evolutionary virologist. So he's like, "This looks pretty weird. It doesn't necessarily look natural." He called up Tony Fauci, told Tony Fauci that he thought this was weird. And so they convened this conference call to have a discussion about it. They ended up writing a paper called The Proximal Origin of SARS-2, which was published in March of 2020. And that said that we still don't really have much information besides the sequence of this virus. But in looking over the sequence of the virus, it actually does appear like it's natural. So after that, this was a hugely influential paper in 2020. After that, a lot of us kind of made that assumption, but kept looking because we still didn't really have any data about the early days of the pandemic. And so slowly, some of this data started to come out. There was a leaked report from the Chinese CDC that came out that said basically they had done this environmental sampling inside the Huanan market, where they basically took a bunch of samples off of different surfaces and things, and then they sequenced them. There was also reports in the WHO mission that happened in early 2021 that talked about some of the animal testing and things that they had done. So this information started to trickle out. Unfortunately, we don't have any information at all about sampling from the actual animals that were at the market, because for a long time they were denying that live animal sales were taking place. And I think one of the reasons for that is that is the exact same circumstances under which SARS-1 emerged. And pardon me, there was a dog next to me who is just kind of whining. She wants attention. And I'm talking to my phone. And anyways, so we don't have those samples like from the actual animals that would prove that the animals were infected. So we just had to do a lot of detective work to try to figure out where everything was in the market, looking through old documentary records of things like people getting fines for selling animals, old like shopping lists basically of what the various stalls sold. And then we also had a secret weapon, and that is my co-author, Eddie Holmes, who's an evolutionary biologist at the University of Sydney in Australia. In 2014, he had visited the Huanan market with several of his colleagues in China who said, look, this is the type of place that a pandemic could emerge from because look at all these animal sales. Eddie took a picture of a raccoon dog in a cage with some birds next to it and a variety of other animals that were all being sold there. That stall had 5 environmental positive swabs for SARS coronavirus 2. So it's possible, we don't know which stall it was, but it's possible that the pandemic could have actually started at the very same stall that Eddie took a picture of back in 2014.
First of all, if you're Eddie, you've got to like be living in a bizarre world where you kind of predicted this, and this is literally what was led where the clues led back to. Someone did give us another comment about the motion sickness. So I don't know if you can grab like a copy of a textbook or something like that or something. I don't know.
Yeah, let
Um,
me just set it
yeah, I'll
here
I'll
and
let
try to
I'll
figure this out.
yeah, I'll let you do that, and then, but you know that's what I think is so interesting. If you look at I'll summarize a document that I finally read, which was the U.S. Congress had a select subcommittee looking into the pandemic origins, and this was run by a bunch of Republicans who kind of already decided ahead of time what they wanted it to say. And I finally read it, and basically what that most of that document is a lengthier description of the emails that you just described, which is that Christian Anderson said, "Oh, this looks a little fishy," and then Fauci and his friends and Collins were like, "Oh, let's get everyone together and talk about this, and make sure that if that's true, that we know. If it's not true, we got to stop that from getting out there." Um, and basically, Anderson changed his mind. He's like, "Yeah, now that I look at it a little more carefully, yeah, it's pretty much there's that dog won't hunt, and that's fine. Let's move on." And the fact that he changed his mind seems to be the proof that the Republican-led committee has. Um,
Sorry,
it's
dog
okay.
again.
Yeah, yeah.
My ring light broke, and so I don't have a tripod, which is
It's
why
okay.
this is
All right.
happening.
I'm gonna
Sorry.
send you a tripod, and that's fine.
I've
Uh,
already got one ordered.
okay. Okay, that's good though. So anyway, going through that document is basically a forensic analysis of the emails during which Anderson changed his mind, and there's really no engagement in that document with the incredible genetic linkage and heat maps that are in the papers that you've been a part of. And I've just always said that you can always tell a little something about the situation, not entirely, but you can tell a little something or maybe a lot about the way people frame things. And to me, I think that you and I might have come to this question with a sense of agnosticism, meaning if there was a lab leak, let us determine that and let us absolutely figure out how to prevent that from happening in a way so that we can actually get ahead of the next pandemic, not certainly don't cause the next one, and make it better. There's no real downside to it having been a lab leak from our perspective. It just isn't the case. Whereas the people who are absolutely insisting that it's a lab leak kind of want to use it as an excuse to light the entire industry on fire and say, we're not going to do pandemic research anymore.
And that's exactly why they're doing it. It actually doesn't have anything to do, I don't think, with the origins of the pandemic or that people actually even care who are saying this. They're saying it as a pretext to cut this whole area of research. And that's become increasingly obvious to me with this current administration. And Jay Bhattacharya in particular, has been tweeting that it's a lab leak since 2021 and he called me a reckless virologist way back then who doesn't care about biosafety, which is absolutely not true. But he's been using this line for a long time, and it's really to undermine the conventional processes of science, which he does not follow. His idea of, quote, gold standard science is actually quite unscientific because in that version of gold standard science, you approach a hypothesis with the idea that you're trying to prove it. A real scientist makes a hypothesis and then tests it. And if it fails, you make a new hypothesis. It's not considered a failure. Like that is actually the scientific method. And so Christian Anderson changing his mind is just one example of the scientific method in action. He did a little more analysis, he did a little more research, and he discovered that, oops, my original hypothesis was probably incorrect. And one of the things that sort of motivated this group of co authors to come together is we were getting really, really tired of explaining this over and over to people. And certainly from my own perspective, I was really defending the profession of virology in the sense that, first of all, we can't just throw in a furin cleavage site and juice up a virus, like just because we think it might work. That's not actually how virology works. We're much more careful with that. We're not just doing experiments to prove to each other that we can make more dangerous or transmissible viruses. Um, the other thing for me was that this virus didn't look like something that a virologist would have made. Some of the features that have been pointed out, like the furin cleavage site, for example, are suboptimal. It's not the kind of, why would you use that furin cleavage site when you can just toss in one of the furin cleavage sites that's in the spike protein of MERS coronavirus, for example? And so a lot of this didn't make sense to a lot of us. We were kind of trying to educate people about this. And we sort of all came together in frustration and we said, we're all scientists. Let's start looking for ways we could actually test this hypothesis with real evidence that we can verify. And so that's kind of how all of our work started together. The Zooinati is what some of the lab leak people call us because we're like the Illuminati, except for zoonosis.
Right. The idea that a zoonotic infection, for those of you who don't have a PhD, is that the source of a virus came from an animal in the
Right.
wild, a quote unquote natural source, as opposed to, say, a lab generated accident. Um,
Correct.
and I think the reason I wanted to start here with you today on this was that the nature of this piece that we debunked basically in our Substacks really at the core f- is driven by the conviction that the COVID-19 pandemic was caused by a lab leak. And if you hold that worldview, my goodness, a lot of things would follow from that. Now, first of all, some things could follow from it that are perfectly productive. But if your viewpoint is it came from a lab and the same thing's going to happen over and over and over again, and the more lab research that we do and that we support, the more bullets go into that roulette chamber, so to speak, with respect to a analogy about this, and therefore we should not do that. And this strikes me as what I wrote as the head in the sand approach. And there's really a couple things they don't want to do in terms of preventing the next pandemic slash finding ways to mitigate it when it happens, because those things can actually go hand in hand. One of the things they don't want us, the scientific community, or you, the virology community, is to actually go out into nature, into caves, looking for animal viruses that have potential to cause disease in humans. In other words, they're saying, look, we already know that flu and coronaviruses are the things that they cause pandemics. Why go find a bad one? You're just flirting with disaster. How do you respond to that?
Well, I get told that I'm asking for it all the time, whenever I say anything that's remotely controversial. So, to me, that is just a completely ridiculous argument because in reality, this was never the US pandemic plan. And I think it's important to actually start there when considering this as this used to be the US pandemic plan. The US pandemic preparedness plan was a lot more complicated than send people into bat caves. They can find viruses. Then we bring those viruses back to the lab and juice them up. And then somehow Big Pharma gives us a bunch of money because we're also making these bad viruses so that we can develop vaccines for them and get rich, essentially. is the idea. That was never at any point a US pandemic preparedness playbook or plan or anything. This type of research, virus discovery research, and then countermeasure development, trying to figure out what the next pandemic might be, is actually a really important part of pandemic preparedness because it's obviously a lot more helpful if you know what to look for. You're trying to do surveillance, you're trying to monitor its potential for emergence. You want to try to rank viruses as like this one's dangerous, this one's not. And unfortunately, the only way that we can do that now with our current technology and our current level of knowledge is to actually go out and find viruses and try to figure out which of those is going to be more likely to infect a human, which one's going to be more likely to transmit between people and cause disease in them. So, this work is important for pandemic preparedness, but it's not the whole game, not by a long shot. I mean, as you probably know, being a clinician, one of the biggest issues that we had during the pandemic was the resource load on the healthcare system. And so a lot of our pandemic plans had to do with that. Like they had to do with making sure that healthcare resources were in the right place, making sure that infrastructure was built out. It had nothing to do with this type of work, which is important virology research, but it's not the only kind of virology research. And it certainly wasn't what we were counting on to protect us from the next pandemic.
Yeah, and from the clinical side and from the epidemiological side, which is where I spend a lot of time, this actually healthcare capacity, which is not addressed in their document, but healthcare capacity is not just a matter of concern to Health and Human Services, HHS. It's actually a matter of national security.
Yeah.
So I used to get phone calls when I was doing some modeling about health hospital capacity. I was getting phone calls from the Department of Defense and from Homeland Security and from the Veterans Administration, because these people were saying, wait a sec, if this Omicron thing is as bad as you say, are we going to get in a situation where the hospital are going to be full? And then what happens if anything else happens? What if there were a bio attack on top of that? Or how's it going to function? And so the idea that you can just kind of wait around it doesn't work. And on top of it, this is the same administration that's making changes that are actually going to lead to the closure of critical access hospitals, so that when another outbreak happens, we won't have the kind of capacity that we even had in 2020, which actually wasn't adequate. Let's kind of pivot to the next big thing that they're very afraid of. And again, I approach this with humility. I've done lab research. I spent several years doing virology and immunology. So I know that it's difficult work. I've worked with HIV, which is not nearly as dangerous as a pathogen as some other ones that are out there that we work with. But they're very, very afraid of what they call dangerous gain of function research. And they sort of refer to that as an entity. And I had never really seen it framed that way. So first of all, just repeat for everyone is what they mean or what they think they mean. And how do you think about gain of function research and both its utility and its risks?
Yeah. So I actually think of gain of function research by its more traditional definition, which is when you're doing cloning, you're doing molecular biology, you do something that makes something else gain a function that it didn't have before. That's a gain of function experiment. This is a term from genetics.
But can I just
And
even right there stop, because this is where we get into taxonomy very quickly. You can create a gain of function, meaning let's say, okay, I'm going to give this pathogen a new protein or a new version of the protein, and what that does is it changes the way it cuts itself up. That could actually lead to a less effective virus. So my
correct.
gain of function could actually lead to a loss of function, or the other way around. I could say, hey, I'm going to actually take away this thing. And it turns out that through science and through that experiment, we learned, oh, you know what's interesting? We were surprised. This actually increased its ability to do something else. And so what I thought was loss of function is gain of function. And so is there such a way to distinguish this in the first place?
Yeah, so that's what I was saying. This is why this is such a confusing term because it's largely a scientific term. And I could do a gain of function experiment where I put GFP, which is a green fluorescent protein. It's a protein that makes cells turn green into a bacteria that's completely harmless. And now it's been a gain of function because it's green. That is not what we're talking about here. And I think that's why they added the D with this new administration, that's dangerous gain of function. But the way this was actually regulated before, I'll briefly go over the history. In
Yeah.
2010, 2011, two flu virologists did an experiment. They each did experiments where they adapted H5N1 for aerosol transmission between ferrets. And they published this, or one of them talked about it at a meeting, and everybody said, "Oh my God, holy shit. Like, they are making bird flu go airborne." And that is a gain of function. And so we need to regulate this work because it's very dangerous. And there was a huge debate about, like, should they be allowed to publish their methods, like that kind of thing. They ended up publishing their methods because these methods are the same that you would use to do a non-dangerous gain of function experiment. And one thing that for everybody to keep in mind is that the risk assessment for every single experiment in virology is going to be different. It's going to depend on the pathogen. It's going to depend on what you're doing to it. It's going to depend on the host. It's going to depend on your lab facility. So you can't really say, like, oh, all of these things are gain of function, they're
Right.
dangerous, and we're going to ban them. You have to take it experiment by experiment. And you have to have the technical expertise to be able to accurately assess that. So that's what they tried to do in 2017. They came up with the Potential Pandemic Pathogen Care and Oversight Committee, or the P3CO, it was called. And that defined gain of function research, essentially, or they called it enhanced potential pandemic pathogen. That defined it as taking a virus that exists and effectively making it either more transmissible or more pathogenic, causing more severe disease in humans specifically. Now, this was difficult because the language said that it's reasonably anticipated, because obviously the only way that you could actually test this experimentally is to infect a human and see if it's more transmissible or more pathogenic. And, you know, reckless virologists don't do that either. We don't do human challenge studies with new pathogens that we don't understand. So, there's a lot of debate about that reasonable anticipation language. And Jay, since 2021, and discovering that he's a lab leak guy, has really been saying that this approach is completely inadequate. We need to ban all of this type of work. It's completely unsafe, and all the next pandemics are going to come from labs if we allow it to continue. Now, fast forward to the presidential executive order on improving the safety and security of biological research. And that started using this dangerous gain of function language, where the definition has expanded considerably to include, depending on who's sort of interpreting it, it could include almost any virology experiment with almost any virus. And so that essentially, by using a non-specific, non-precise term like dangerous, that's in the eye of the beholder, how dangerous it is. And Dr. Bhattacharya, is not a virologist. He's a health economist and an epidemiologist. He is not actually qualified to judge, like, what would be dangerous and what would not. Now, Matt Memoli is an influenza virologist, and he would be qualified to assess this. But I find it very interesting that he apparently didn't do these type of assessments with his own work that he has published prior to becoming the principal deputy director of the NIH. It seems that he's had a big change of heart since joining the NIH, where now his definition of dangerous gain of function research is basically research that anybody except for him and his friends are doing. So, it's very frustrating, because this is actually not a technical or scientific conversation that we're having anymore. And DGOF, dangerous gain of function research, is not a technical term. It's one that's been made up by this administration in a presidential executive order.
And I want to dig in a little bit on that last point, which is that Dr. Jay Bhattacharya, that's the NIH director, his principal deputy director, Matthew Memoli, who was the acting director prior to Jay getting confirmed, he was sort of, let's say, picked from obscurity out of the NIH. He was an NIH intramural researcher, meaning not like an NIH funded researcher at some institution, like whatever, at University of California or something. He was the NIH actually does research in house. And he
Yes.
had a career sort of postdoc. I'm not sure if he was a PI, but he was sort of, doing some reasonable work over the years on influenza. And to your point, what he was doing in influenza vaccine research could easily be described as dangerous gain of function, correct?
Yes, that's correct. So, Generation Gold Standard, which is the inactivated whole virus flu vaccine that has been proposed as an alternative to mRNA vaccines. This is very ancient flu vaccine technology. Essentially, this is the original way that they made inactivated vaccines. So, they made this what they're calling a universal flu vaccine. And I was very curious when this announcement came out. So, I looked up the one paper I could find about it, and I see that they vaccinated mice, and then they challenged them with a bunch of different flu viruses. And I look at what those flu viruses are. Fully reconstructed 1918, which was a virus that didn't exist anymore, essentially in the real world until the current acting NIAID director, Jeffrey Taubenberger, resurrected it with cloning. So that is definitely a gain of function because the virus wasn't around, and then it's around now.
I mean, one of the deadliest viruses in human history.
Correct, and I fully support actually reconstructing 1918 because I think that it's really important that we study known pandemic viruses to understand why they
Yeah.
became pandemics. So they use that to test it. So that's a gain of DGOF virus, and then they had all these figures with what was listed as a chimeric avian influenza virus. And without going into all the details of how these things are made, they basically did gain of function research to select for influenza virus reassortants. These are viruses that they made, again, that were deadlier. So they literally selected them on their basis to be DGOF pathogens. Um, and I was shocked when I saw that because I was like, wow, they hate gain of function research, but have no problem using its products to test the vaccine that they're trying to push that Memoli himself essentially awarded to himself for $500 million.
Yeah. And what Dr. Rasmussen is referring to right there is that after Dr. Memoli served his brief tenure as the acting director of the NIH, having been someone that people had heard of, but certainly not next in line to be the acting director during a transition period, he was rewarded for doing the administration's bidding by in really two ways. One, he was elevated to principal deputy director, which basically makes him like the right hand man to the director. It's the literal number two at the agency, and $500 million in research intramurally, which is an unprecedented number. And I believe we're talking on the order of magnitude. You don't even hear about $50 million at a time getting promised to any project inside the government. We've heard of $500 million contracts going for like, okay, for Moderna to spend many years on a variety of mRNA vaccines, but for that kind of cronyism was really appalling. And it wasn't that action was not included in the whistleblower complaint that two former NIH leaders, including Dr. Jean Marazzo, have filed, but it's a big part of it. I've spoken to Dr. Marazzo about this, and she's been quoted on the record that part of her concern of them getting rid of career scientists is that they're replacing them with people who are sort of bending the knee to the administration, and in exchange for that, they're getting $500 million of research. I
Yeah.
mean, Angie, what would you do with $500 million of research funding?
A lot of good science that I've wanted to do, but it's really hard to get grants because the other thing is there's not enough grant money for everybody to just do what they want scientifically. So that's why it's so hard to write grants. You have to actually write experiments that are well designed, that solve a scientific problem that is going to make a big impact. And you have to show that this is a significant problem that is going to have a big impact. That $500 million award for Generation Gold Standard, as far as I'm aware, it did not go through any type of scientific merit review. So we write a review or a grant, it goes to a study section, which is basically a bunch of NIH funded researchers that have expertise in the area, and they review it for scientific merit. It's a group of people. It's taken very seriously, and then it's overseen by NIH staff, and there's a longer process to it. But the basis for awarding grants is that you can show that this work has merit. It's going to lead to an increase in advance in our knowledge, and it's going to actually benefit American health. This did not go through any of those processes. It was essentially cronyism, like you said, it's a payout.
Yeah, and I could sort of see a world in which you would support this research at the level of $5 million or 10 or something like that. But for it to have been a $500 million commitment was sort of an audacious confession of what is to come. But
Yeah, on
I
the
do
basis of essentially one paper.
yeah, and let me ask you just further. They did not prove, obviously, that this universal flu vaccine based on a truly a 20th century technology. It's not a particularly novel idea. It's actually an old idea, but they did not prove that that is adequate. So, in order to continue to pursue this universal flu vaccine using this older platform, they will indeed have to continue to test it on dangerous viruses. Otherwise, they will not know if it works because the only other way to do it would be to infect humans, and that cannot be permitted.
Right. And you would think that they would have thought that through, considering how upset they were in that piece about drugs being stockpiled and vaccines being stockpiled that have never been tested in humans before.
Yeah. My second question about this is what do they imagine virologists should be doing in their spare time to prevent the next pandemic if it is not to actually do the kinds of tests that, frankly, Dr. Mamoli is doing and that virologists do all over the world every day, which is to assess potential novel viruses for their ability to cause disease and to be transmissible among animals, and therefore to extrapolate to humans, and then hopefully we can, have a head start on the next pandemic. What do they imagine research would look like?
I think that they imagine that we're not going to be virologists anymore.
Yeah, that's.
We're going
I
to,
was worried. I was worried you'd say that shit. And um,
I think they'd love to see me waiting tables somewhere, maybe working in like an old lady strip club.
great. My other question is about the platform. The mRNA platform is, there's nothing particularly special about it in terms of its magic, except that it has a couple of advantages. One of the advantages of it is you can kind of cook it up in using laboratory equipment without the need for longer processes like growing the virus or growing the vaccine out of actual viral cultures, right? And so, can you explain to everyone how, if we, for example, were to rely on a vaccine platform like the one that the NIH Deputy Director Matthew Mamoli prefers, or anything other than mRNA, how many months longer would it take us to get a vaccine if a pandemic were to occur right now?
So for a flu vaccine, we actually know this pretty well. And I will say that inactivated vaccines, whole virus vaccines, that was the first COVID vaccine that was made available. It was made by China. Because in some senses, it is easy to just grow up a virus, inactivate it, give it to somebody, and then voila, you've got a vaccine. Then you don't have to find out like what you need to make the mRNA encode, like all that kind of stuff. But you're right. For a flu vaccine, mRNA would be much faster, especially if it was a brand new strain of flu. Because the way that we make flu vaccines now, they are inactivated vaccines, is we grow them up in eggs. So we have all these flu viruses that the WHO Collaborating Centers for Flu maintain. When a new virus comes out, for example, the H5 virus, they made 10 million doses for the US Strategic National Stockpile. They matched the closest virus in that library to the virus that was circulating in American dairy cows, and then they made the vaccine out of that. Now, you can make 10 million doses in a fairly short period of time, but a lot of influenza vaccines, the majority of them in the US, are actually grown in eggs. So, embryonated chicken eggs. So, you need that many chickens. Now, for flu, especially if it's avian flu, guess what species that infects? Guess what it causes? We had an egg shortage last year. Egg prices are still high. So you actually are even more screwed with a flu vaccine if it's bird flu, because you might not have enough eggs to manufacture that quickly enough. But it would take essentially for to make that H5N1 vaccine, if it does become a pandemic virus, it's going to require, nationwide doses and then some. And, you're talking a year, 9 months at best, but you're talking a year production time to actually make that many doses. Where we know from the Moderna stuff, you can actually get through 3 phases of clinical trials in less than a year and still manufacture vaccine at scale.
Yeah, I'm actually curious. I don't know the answer to this. I know that the way that the phase one and then combined phase two and three trials for the COVID vaccines, the way that that proceeded was obviously record setting. It was warp speed and all of that stuff. But I have looked at sort of some of the power calculations and some of the assumptions about the epidemiology that in retrospect, we actually knowing what we know now, we could have gotten it actually even sooner. And I don't say that as like a criticism of the scientific community. I think that there's some limit just based on safety tail. Like you can't say, "Oh, I've got this vaccine right now, and I'm going to give it to people in a month from now, see if it worked or not." You need to show at least several months later that these people aren't coming down with severe side effects. Safety comes first, and there's some. So whatever your minimum safety window is, that's the actual theoretical fastest. So even if you could somehow prove that this thing was 95% effective after a month, you actually still need 2 more months just to make sure that before you give it to 300 million Americans or more, that they didn't all drop dead from some unexpected side effect. So that's like kind of the theoretical. But let's imagine that we've done a really fast phase 1. We got our phase 2 and 3 data like in May or June instead of in November. How long would it take Moderna or Pfizer or BioNTech to get 100 million doses or 300 million doses out the door?
I mean, it would. I'm not sure actually about the number of doses, but you could essentially start manufacturing that day. They're very fast to make, and you can really, really easily adapt them. It's a matter of essentially typing stuff into a computer, and then going through your existing manufacturing process. So
And
they're fast.
And the ingredients aren't expensive. The
No,
expensive part is the research and development, right? What
right.
you're paying for is the fact that it took them, 10, in some case, 20 years to be able to do what they do, correct?
Correct. And actually, I mean, that's another good point that we touched on in the article is that we wouldn't have been able to make those vaccines so quickly if it had not been for the NIH funded basic research on SARS coronavirus 1 that allowed us to see that we knew which antigen to target. We knew, that the mRNA vaccine platform worked and was safe. We were able, because of that foundation of research that had been funded by NIH, which they would like to cut, that's how we were able to get that out the door so quickly.
All right. So if Jay Bhattacharya were to call you on the phone tomorrow and say, "Hey, Dr. Rasmussen, I really need your help," what would you say?
I'd say, "Sure."
No.
Actually, I a lot of people think that I'm just gonna be like, "No, fuck off." But no, if somebody from the government calls me, it doesn't matter what their political beliefs are, like, I do think it's my duty to try and help them. I don't think that Jay would ever call me and ask me for my help, and I'd probably have a heart attack also if that happened because I would be so surprised. But I would try at least to help him understand, like, what he's got wrong about all of this stuff. I don't think also that will happen, though, because I don't think that this article was written in good faith. As I said, the entire premise is essentially a lie, that this was the pandemic preparedness playbook, and it was not. And when you're dealing with somebody who is that willfully, I think, dishonest and is misleading people, it's very difficult to help them when the method that you would use to help them is by helping them understand what the truth really is.
And I'll just add that there was there were some other publications out of HHS that basically said, look, the reason that we're not pursuing mRNA technology right now is that the trust is just so violated there. And therefore, we just can't do it. And my whole feeling is, well, I think pretty much those are the same people who actually sowed that mistrust. So that's a little bit like throwing a rock at a window and saying, hey, your window's broken.
Yeah.
But also, if you truly take the scientific approach, which you outlined earlier, it comes from a place of humility and saying, "We don't know." It could be that my particular favored approach to the next vaccine or the next pandemic is not the right one. But I think that as they say, "Por qué no los dos?" or why not all? When you have this is the argument for public investment. There's no profit model for taking a bunch of low risk pathogens and a bunch of possible therapeutics or vaccines that probably won't be useful, and trying to convince investors that not only will this thing emerge to be important, but we've got a solution for it. It's way too risky, and so the government can come in and give funding for things that are low probability but high stakes.
Yeah. And that's exactly the situation. Like, you look at stuff like Ebola, bird flu, very few people actually get Ebola, but it's such a severe disease that it's really important to study it because when there is a big outbreak, as we saw in West Africa about 10 years ago, it's absolutely devastating. And it's devastating to the economy as well as potentially to agriculture, depending on the pathogen. certainly that's the case for H5. But we spend money studying the things that we think might cause us a huge problem. And even if they don't, something very much like it might come along and cause that problem. And I just want to comment very, very quickly on something that you said, like, why not both? This is something that has been really striking to me because the premise of all of this is that pandemics only come from labs. They never come from nature. But
That's
in
their that's
reality,
their that's their stated fantasy. I mean, I just wanna say that
yeah,
that's their idea.
it's not remotely true. And I mean, this is exactly that distrust that you mentioned. There's never been any evidence shown for where this distrust comes from. I would argue that it's coming from lies like that, where you're essentially saying that, oh, SARS-2 couldn't, it had to be a lab leak. So every other pandemic has been a lab leak, including things like 1918. I don't know why they didn't even know that flu was a virus then. They didn't even really fully know what a virus was. But pandemics mostly come from nature. They are mostly zoonotic viruses that come from animals in nature. And by not doing anything about those pathogens and only focusing on labs, which there's been one pandemic that might have been associated with research, and that was the 1977 flu pandemic, which was probably not a lab leak, actually. It was probably the result of either a human challenge trial that got out of control or a vaccine that wasn't fully inactivated. But in any case, like, there have been almost no pandemics started by lab leaks, or no pandemic started by lab leaks. Only one pandemic may be started by research. Everything else is natural. So you're telling me that it's a good strategy to only focus on shutting down this research we don't like, which, by the way, we need to deal with the natural pandemics, and not doing anything about the natural pandemics. That
And
makes us so much more vulnerable.
and I think that it's, what I find is that people who aren't lab scientists or haven't done lab work don't work in our ecosphere. I think it's important for them to hear from us that we know that lab safety is important and that lab leaks actually can occur and they do occur, and that does not necessarily mean that any of them are serious. Can you talk people? I think again, just the transparency and the authenticness of saying, you know what, yeah, it's true. If you drive your Porsche 130 miles an hour down the wrong way, you'll die. But if you drive it under the speed limit and wear your seatbelt, you'll probably be okay. But so it's okay to say, like, can you tell us, like, what are some legitimate things that you worry about in terms of like lab safety, and how can we reassure the public that that's on our minds?
The number one thing I worry about whenever I go into the containment lab is lab safety. And there's a very good reason for that. I don't want to infect myself with something that is a deadly virus that there is no vaccine or drug treatment for. I don't want to infect any of my colleagues with that. I don't want to infect anybody in my family with that. I don't want to infect anybody in my community with that. I don't want to die from one of the pathogens that I work with because I was careless with it. And so, for that reason, I mean, when I'm training somebody in containment, I do not let them go anywhere near the containment lab if I think for 1 second that they are not taking that risk seriously and putting it above all else. My number one priority when working in the lab is biosafety. The experiment has to be scrapped for the sake of safety. We'll do it again. If the experiment's too dangerous, we're not going to do it. Um, it is very, very heavily regulated in addition to that. So it's not just me being like, oh, I'm careful, but my other colleague over there is not careful. No, this is regulated by federal law, and I work in Canada, but I'm originally American, so I'm familiar with both systems of regulation. You cannot mess around with what's called a select agent in the US and just, hope for the best. Like, it is a federal law that if you mishandle this pathogen, you are going to jail. Actually, there are criminal penalties for it. You have to get a security clearance in both countries to even be allowed to go anywhere near these pathogens. You have to train for years, essentially, to be trusted with these pathogens and to be competent in handling them safely. And that goes down to even the smallest thing that we do. If I'm disposing of garbage, it has to be done absolutely according to protocol in the most safe way possible. And we have multiple redundancies throughout our facility. I can't really get into all of them, but every facility has this.
But I
You
think that,
build in all these levels of safety because it literally is considered the most important thing that you do when you're doing virology research.
and I want to just take people in the lab for a minute. I was actually just reading some Susan Sontag about how we understand pain, and she basically took a few chapters to say what boils down to a picture is worth 1,000 words, that war photography had a big impact on how we see suffering. And I think that sometimes we miss that opportunity in COVID because, quite frankly, of HIPAA. We can't like just show dead people.
Yeah.
And then, and other times just not showing people what it's like. But I also think that it would be really instructive for people to see the the extent to which, like when I was a researcher, what I had to do when I was handling just some HIV virus, which is not airborne, by the way. Like the odds of me like giving myself HIV, even if I wanted to, would be very, very difficult in that lab. But even so, you had to go in the hood where the negative pressure and everything. You're wearing, all this protective equipment, and so for people to imagine that a lab leak would occur, it, it, that's based on a lack of understanding of just the pure infrastructure. It's like the reason that 3-year-olds don't crash your car is because 3-year-olds can't operate your car.
Right.
And so, right, it's not like because, oh geez, we just watched them. No, the infrastructure is too complicated to actually have the risk. So I wish people would kind of see that.
Yeah, I mean, I think it's spoken, like I said, Emily knows that. He should know better than to have written something like this because working with high path flu, which there's an exemption right now because of the H5 situation, but it is a select agent. In fact, it's an overlap select agent, which means that it's regulated by both the CDC and the USDA. So you can't just go into the lab. It's not like I get off the phone with a bunch of my virology buddies and it's like, "Oh yeah, guess what I'm going to try? Like I'm going to make an Ebola virus even worse." It's not like I can just go do that experiment for fun like because I feel like doing it. There are a lot of rules in place to prevent that from happening, and by the time you get to the level where you actually have the skills to do that, you would have been screened out of this profession if you were not taking that with dead seriousness as the most important thing, again, that you are worried about.
I want to close with two more things because we're coming up on an hour. One is we've covered this a little bit, but Dr. Mamoli and Bhattacharya seem to conflate conflict of interest and expertise. And RFK has done this with the Advisory Committee on Immunization Practices at the CDC. The idea that if you have spent your life studying something, that you are conflicted by it because you are paid to go to work. And that to me is absolute effing nonsense. That yes, the pharmaceutical company has a vested interest of some of these things, which is why people like me read their papers with a fine tooth comb and call them out on their bullshit left and right, and
Yeah.
celebrate the times when we actually agree, like the COVID vaccines, where we say, "Hey, look, industry actually is correct. There's no concern here. Let's go." And then when they start overplaying stuff, which I think they've done with some of since then, we go back to the usual skepticism. But they have this idea that expertise is a problem. Please respond.
Yeah, so this has been going on and it's been very frustrating in all of the gain of function conversations because a lot of the people who are very anti gain of function work and are very concerned think that virologists really shouldn't have a seat at the table because we're incentivized to get all these grants. And there's this whole separate myth that Tony Fauci was also profiting massively off of this and was committed to biodefense. And so it was just like making it rain grants on people who would do this type of work. And that's absolutely not true. None of that is true. Most virologists, including myself, don't actually do dangerous gain of function research or don't actually do what would have been defined as gain of function under the old P3CO. So most of us don't even do that type of work, even though we might do work that requires biocontainment or anything like that. Most of us like wouldn't have benefited from that even if it were true. But the reality is, I think the reason why a lot of this expertise is being regarded as conflict of interest is because it's very convenient for getting rid of inconvenient experts who will tell you that you're wrong when, again, you're trying to advance an agenda rather than trying to get to the bottom of a scientific question or make sure that you're regulating science in a safer and more smart way.
All right. I do want to end with where they ended, which was I've noticed this very creepy thing that's in a lot of publications coming out from HHS leadership right now, which is that at some point, usually at the end of their essays or their position pieces, they have to sort of bend the knee, kiss the ring, lick the boots of Maha and say, if only we were just a little healthier, pandemics wouldn't be a problem. And geez, if it isn't red dye number three that is the real problem, or obesity, which is a real thing. And I want to point out that I actually have written about this in response to really important research that absolutely shows that one of the best ways we as Americans could have had a better outcome from the pandemic that we just endured would have been indeed to enter it healthier as a population. 100%, like the hemoglobin A1C level of the country, which is the marker of diabetes. I'm sure that for every decimal point it is raised, there is a mortality correlation for a pandemic because pandemic viruses or pathogens can kill everybody. They can, but they're very often going to target people with medical risk. So 100% we agree that we should be making the population more healthy, but don't you it it seems like a non sequitur. How do you as a virologist interpret that part of their argument?
Well, I mean, to me, it was just ridiculous when I read that argument because, like you, I agree that, of course, it would be better if we were all healthier. If there was a higher level of health in the population, no question, we'd have better outcomes. But, I think of 1918, which killed a lot of able bodied young adult people, people who were not sick, people who were not in a typical flu risk group, which is children and older people usually. And viruses don't care often how healthy the host is. They care if they can get into the host cells, if they can replicate once they're in there, if they can be passed on to another host, because viruses can't reproduce on their own. They're obligate parasites. They have to have a host or they die. So, from a virus's perspective, and it doesn't think, it's like basically a biological computer program almost.
Yeah.
A virus just wants to make more viruses, though. And that's all they care about. They don't care if the host is healthy. They don't care if they cause disease. They just want to make more viruses. And so, being healthy, sure, it's great to protect against disease for some viruses, but not all. You actually need something more specific, like a vaccine that will prevent you from getting sick if you do get infected, or a vaccine, even better yet, that might prevent you from getting infected altogether, or at least seriously reduce your risk. Antiviral drugs that you could use to treat severe disease, things like that. We need to have that in addition to being healthier. And I think one of the things that I've observed with the MAHA movement, and I think this is why they always go there, is that the gateway drug to all the anti vax stuff, all the anti science stuff, is really nutrition. And like, let's get everybody healthy. Like, who's against like better food? Nobody's against that. And so that's a way to hook people, I think, and bring them in and then start introducing them to some of these more unscientific and radical and harmful ideas.
Okay, well, I'm going to wrap it up here. And first, what I'm going to do is tell everyone who's been watching this just thank you because we're getting a lot of support and comments, and we appreciate the respectful dialogue. And it just means a lot because I work clinically. Dr. Rasmussen is in the lab, and sometimes it can feel a little dark. So thank
Yeah.
you all for like giving us wind to do this work. I cannot more highly recommend Dr. Rasmussen's Substack, which is called Rasmussen Retorts, right?
Yes.
Rasmussen Retorts. And if you're watching this, you know that my Substack is Inside Medicine. And honestly, people always want to know what can they do. And the number one thing they can do is actually leverage these platforms to get the word out. The other side is really good at that. And I think that our side is getting better. But I just invite everyone here to like when this goes live on our Substack later with the summary, like share the hell out of it. Because what our goal here today was just to try to talk about some really complicated things as clearly as possible. And maybe we got to the right level, and maybe sometimes we misjudged. I don't know. But what I can say for sure is, if you're watching this, you can tell how much both of us care about this. And I think that message is actually so important, it gets missed. Like, that we really care that there's not another pandemic.
Yeah. Yeah, I mean, that's it. I thought that COVID was going to be really bad, and now I look at the situation in the US, both with healthcare and with the state of science, and we're going to be so much worse off if we have a pandemic now in the near future. Much, much worse off. It's going to make COVID look like a picnic. And I really can't think of anything that's more important than to speak up for public health and for actually trying to make things better rather than trying to make things worse to serve, a different agenda than keeping as many people as safe and healthy as possible.
And I'll just say that, as someone points out, when does a pandemic end? Who's to say we're not still in one with people, the suffering that we see? And that's right. The tail of these things is so long, which is why it's so important to get it right. And obviously, we're not in the same emergency that we were in several years ago, but it's still, people are still suffering, and that's why the stakes are so high. Okay, thank you, Dr. Angela Rasmussen, for not only just being an incredible researcher, but just generally being a badass human being.
Well, right back at you, Dr. Jeremy Faust. And thank you so much for inviting me to collaborate with you on this. It's been fantastic working with you. And I know that we'll do so again. I
I
think we're
I
a great
I
tag team.
I just hope that it's not that it's voluntarily, that we're not working together next because someone finally figured out that we have a huge problem and we need to get a war room of people. Let's let's we're gonna work together before that happens.
Yeah, no, and I mean, in this case, it happened very organically. You texted me and asked if I'd be interested in doing this, and I didn't want to do it all by myself. So I think it,
Yeah.
it ended up working out great, and it seems like people are appreciating it. So it
Yeah,
was
by the way, the original piece that this is kind of the audiovisual version of is a rebuttal to the NIH pandemic preparedness screed, and that's also on both of our Substacks. And I will say it's nearly 10,000 words, and honestly, I think that I contributed some good things, but your contributions are a genuine masterclass and a public health service, the likes of which I've really rarely seen. So everyone should read that, and thank you, Dr. Rasmussen.
Thank you so much, Dr.
All right,
Faust.
cheers,
Dr.
everybody.
Fauci. See you later.
Yes, Junior. Yeah, bye.
Bye bye.